But I think this time, let's just put a quick introduction into the chat. That way we can all see it and say hi to everyone. And that way it goes a bit quicker. While everyone is introducing themselves in the chat, I will just do a very quick update. We have our next symposium scheduled for the 17th of March. So that is in 2 weeks' time. It will be at 4 p.m. So our normal meeting time. And it will replace our Reclone fortnightly meeting. So really excited to invite you all, and we're in the process of lining up our keynote speakers and creating breakout rooms. But if you're interested in volunteering with us as a facilitator or interested in presenting, please do get in touch. I will put my email in the chat as well. So I'll let everyone do a quick introduction, just a few minutes, and then we will hear from Danielle from Peru. I was going to pronounce your university, but I don't think I would do it justice. My Spanish isn't very good. So we'll hear from you shortly.
Excuse me, I didn't get it. I start?
Danielle, if you could give us just a few minutes, that way everyone can see it. If we give just 2 minutes, is that all right?
Sure, please.
Perfect, thank you.
I give you 2 minutes to, sorry, what? I think I didn't get, I give you 2 minutes to what?
So everyone can do a quick introduction in the chat, and then we'll start your presentation.
Okay, sure.
I think we've got quite a few return visitors from last time, so they probably got introduced during the last meeting.
Yeah, this is fair. Well, yeah, Danielle, shall we kick on? You should have share screen capabilities as well.
This is my power of sharing. Okay.
Perfect.
Good. Okay. Thank you so much for the invitation, and I feel this as a welcoming to the Reclone bi-weekly meetings. So I'm Daniel Guerra. I'm from Universidad Peruana Cayetano Heredia. This is our logo up in the screen. So here in Peru, we are facing what almost everybody is facing. There is a shortage of some supplies. So definitely we need to resort to local production. But our biological industry is way back in development. So that's how a project like this one becomes relevant. I'm going to read the title. This is Developing an Isothermal Molecular Detection for SARS-CoV2 with locally produced biological supplies. This is a project led by Mónica Pajuelo, and I'm a co-researcher here. And as I explain later, we have a lot of help in this project with our friends from Chile, Fernan and Cesar. So we are familiar with the extraction of samples. Once you have the nucleic acid from the virus, you need to amplify it in order to give a signal and get diagnostics. So this yellow part is where I'm going to intervene. We aim to have a test that in only 30 minutes with a single temperature, you can have results. So this is the recombinase RPA, the recombinase-based diagnostics. I'm going to explain very quickly the principle of this amplification of nucleic acids. Once you have the nucleic acids from the virus, this is RNA, you need to produce DNA in order to have a good amplification of nucleic acids. And of course, you do this with a reverse transcriptase. In the presence of the right primers and nucleotides, you have the production of the complementary strand, and you have a duplex. Once you have a duplex, and we understand this can all be done in a single temperature, once you have a duplex, how is that we are going to amplify? Normally, you need to melt the duplex with a high temperature so that the primers can bind and produce the chain reaction. But if you add to the sample recombinase, the recombinase will help the primers to invade the duplex, and you don't need to raise the temperature. So the primers will invade and allow the binding of single-stranded binding proteins, and all this complex is a perfect substrate for the DNA polymerase. So the nice thing is that you don't need to increase the temperature to melt the duplex and to lower the temperature to make the binding of the primers. You can do all this in a single temperature, and the polymerase can work. So putting all these reagents in the single tube, you can have from RNA an amplification of DNA. You see, that's the beauty of the reaction, that you don't need to manipulate anything further than just adding the RNA sample. With this, you reduce the opportunities of contamination. You don't need to add anything else once there is an amplification. And also, you don't need to increase the temperature, lower the temperature, which means you don't need an expensive thermocycler. You can do all with a single incubation at around 50 degrees. This has been tested, of course, for many pathogens already. And this in particular is the experiment done by José Luis Málaga, a young researcher from our university, during a stage in the Tohoku University at the lab of Mayuko Saito. So thanks to this result, we see that there is an amplification of the RNA from SARS-CoV2. In the same conditions, you don't get an amplification of the nucleic acids of many other viruses, not even from SARS-CoV-1, for instance, or MERS. Some viruses can be very, very close to SARS-CoV-2 and still you don't have the amplification. So we have specificity. But we want to have this in Peru. So how are we going to put RPA to work in Peru? This nice picture is a good result made in the laboratory of Mónica Pajuelo, the leader of the project. But this was done with commercial supplies. Probably many of this group here are familiar with TwistDX. This is the company that provides reagents kits for working with RPA. You can program this for different pathogens in your lab, but you still would be dependent of the supplies of this company. And we know in the context of the pandemics, we have a worldwide shortage of biological supplies. I think that all the people who is gathered together today will not be difficult to convince that we need to resort to local production. You see the results are very encouraging. Even with 1 molecule per microliter, you have a positive result. So everything starts with an email, and this is me sending a mail to Fernan saying, hey, can you get us these enzymes? Can you help us with this? And very shortly after, we were receiving the backstabs from our friends Fernan and Cesar from the Catholic University of Chile with all these genes for the production of the recombinase, of the GB32 and the polymerase. Shortly after, there was these grant calls. This is a binational program to have funding for Peru and Chile laboratories. And we proposed, why not we go further to the local production of the RPA in Peru, and then the second year in Chile, they have to reproduce our results. In the meantime, the first year, the laboratory in Chile produces the assay based in LAMP, and the second year, we reproduce their results. So this matching of results between two laboratories was gladly received, and we got the funds for starting this project. By the end of in December last year, we got the funds. Also, in the meantime, we wrote to the Open Bioeconomy Lab, and we received in my laboratory these plates from Stanford. We were very happy receiving them, although we were not ready at that moment to start working with them. So we had the backstabs from Chile and the glycerol stocks from Stanford, and we stored them. and we made mistakes on how we store them. So I think it's useful to know what not to do and still what results we could have out of this. In the first place, from the backstabs from Chile, they were stored at -20 by mistake, and luckily they did not die. We were very nervous about it, so there was plated heavily, even exaggerating a bit. But maybe if we didn't exaggerate, we wouldn't have recovered a GP32 clone. Well, these plates don't look nice, but the good thing is that colonies were there. So we have colonies, the bacteria were alive. And from the plates, we got also colonies. They were stored at -70. These glycerol stocks from the plates of Stanford. Now, we have colonies, but we have to confirm if we have the clones we want. And then we made a colony PCR. That's the most efficient way to test many colonies, to our resort at least. We used the fact that they were flanked with T7 primers. So these universal primers helped us to screen many colonies quickly. And these results, to say it shortly, we confirmed the presence of GP32, UVSx, and BSU, the polymerase, but we could not confirm the reverse transcriptase and UVSY. We insisted on this by growing a colony and extracting plasmid to see if we can make a proper PCR and see if maybe the colony PCR was not properly done at the beginning. So we have problems with this, right? With the reverse transcriptase and the UVSY. Insisting a bit, the UVSY was there, good, but the reverse transcriptase, we just could not amplify it. So that's our position right now, that we have the clones for everything except for the reverse transcriptase. With these plasmids, we transform expression bacteria, BL21. We are using BLR, which are knocked down for REC-A. Well, we have nice colonies. And this was done just a few days ago, last week. And today, literally today, not today in a poetic way to say that these days, no, today, this is being done from a colony. We have to make a quick expression trial to see if we get abundant soluble expression of the UVSx. We're starting with that one. So the trial is very simple. It's just to grow a pre-grown pre-inoculum, and then a bigger culture until some density, and then splitting it in 4 for trying different temperatures. Because at 18 degrees and only 4 hours, we foresee little expression, and because at lower temperature, it is common that solubility is better for many enzymes, we also are optimistic on trying the 18 degrees for much longer. So these 4 samples will be run in electrophoresis tomorrow to see if we have abundant and soluble protein of this UVSx. Next, I'm not saying tomorrow because that would be exaggerating, but very, very, very soon, we need to test the activity of each protein. Of course, we should not just put all the proteins together and see if we can have a proper reaction of the diagnostics. We certainly have to test the activity in a quantitative way, a reliable way of each batch that we are going to produce. And for this, very open to suggestions. At this moment, we are in the level of ideas, browsing the literature, seeing sometimes very old articles from 1976 about the activity of GP32, how to test it. It's not an enzymatic activity, it just binds the DNA. And of the recombinase, and of the reverse transcriptase, and the DNA polymerase. Of the transcriptase and polymerase, we can compare the production of DNA, and we can compare it with the commercial versions. With these other proteins, it might be a bit more difficult. Of course, there is a commercial version of GP32. I'm not aware of a commercial version of UVSx, and why? Probably we could get RAD51, the homologue of humans. And well, just to quickly mention these ideas that we are going to try to assess the quality of each batch of production. is that for GP32, we are going to base our observation on the fluorescence of the tryptophan. So GP32 has 5 tryptophans in its structure. They are partially buried, and you see a blue shift. This is a soluble tryptophan, and this is the tryptophans from the GP32. So you see that there is a blue shift. So they are partially buried. Still, they fluoresce a lot. And if you make a spectrum of fluorescence, so this is the emission spectrum, you see that the protein alone emits much more light than the protein in complex with DNA. So with this, we should be able to calibrate an assessment of the activity of each batch. For the recombinase activity, so there's this paper observing the activity of RAD51, the homolog recombinase of humans, and there is activity measurement by quenching. So when you have a third strand and it invades a duplex where there is a quencher, the fluorescence goes down. Or you can do the opposite. You have a duplex where you have a fluorescence that is quenched, and when you invade it with the third strand, thanks to the activity of the recombinase, then you liberate the fluorophore and you increase the fluorescence. As I said, this is in the level of ideas at this moment. We have to try this. So to design the oligos with the labels and to see if our tools are good enough for the limit of detection these needs. I think it does. The first estimations point to the right direction. So this is the team from Lima. These are Lucero, Nicolás, and Lucero. And Monica is the leader of the project and a collaborator very much about the production of the proteins, while Monica is more specialized in the development of the test. And our friends in Chile, Fernand and Cesar, who kindly provided us with the clones and a lot of advice about how to produce the proteins. And thanks to the funds of Peru and Chile, and to the collaboration of Tohoku University, the laboratory of Mayuko Saito. And many thanks to Open Bioeconomy Lab for the plates and the clones. That's
Excellent.
it.
Excellent. Thank you so much. If anyone has any questions, I think we had one question from Rachel in the chat. I don't know if that was directed at Danielle.
Yeah, well, I think for everyone who's doing RPA, all these reactions, it's hard to know what's priming the reverse transcription. Sometimes, I mean, you should have a 3' end to get the reverse transcriptase to make the cDNA of your target.
Yes. The primers. Okay. So the primers for the reverse transcriptase, you can choose from different places in the genome of the virus. And so this is very nice information that Monica shared with me.
Mm-hmm.
This is the one that we are using. This has been tested in Japan already, and they have been reproduced, these results also in Lima.
Sounds good. Yeah, because I think you can get extra specificity if you just are reverse transcribing what you're looking for.
Definitely. And there's an option also to make it multiplex, but it's not the case. We're only targeting a single region.
I think it looks great. I think using 3 enzymes instead of 5 primers maybe is not, well, I don't know which is better.
We've had troubles with multiple enzymes. Smita can chip in on, like, RPA in general in terms of the production of the proteins has been a challenge to validate because there's so many enzymes. And I was just saying in the chat, like, it'd be really nice to see if those activity tests, if you're able to validate those, because we've mostly been working just on functional RPA assays, as there were other things that we also needed to do in the projects and didn't really get around. But I know, Smita, if you want to comment on your experiences there.
Yes, I agree. As you said, it's difficult to test each protein separately, but we straight away jumped into RPA and it works like straight away. But my personal experience with RPA so far is the concern with GP32, whose working concentration is very high, and in-house purification has been very laborious for me. Of course, it depends on what kind of purification system and how large system your facility has. And also, I have found 2 sources of UVSx and UVSY commercially. I can email you them, although they're not of the same sizes as the constructs we have or has been published. And I have personally not tested them, but the 2 commercial ones as well.
Nice. That would be very helpful. Thank you, Smita.
No problem.
I'm concerned about the difficulties we will face with GP32. We hope that the his tag won't be a problem. I have little clues about some articles problem, but they don't show.
Yeah, so the his tag I think is at the N terminal, although the active site for GP32 is also very close to the N terminal. And the commercial twist DX GP32 also has some mutation around that region to increase its performance, but we are not aware of what kind of mutations they are. Although it is, they have mentioned roughly in their patents, but not exactly what it is. I did initially have problems with GP32, which turned out to be causing like slower amplification. It could be probably where the his tag is located. Maybe a C terminal would have been better. But eventually, I think everything comes down to how pure is your protein sometimes in your mixture. And also for me, the working pH of 8 and not 7.9. 7.9 is what is prescribed, but pH 8 works better in my seem to perform better, especially with UVSY protein, which has its isoelectric point very close to 7.9.
Yeah, that's very interesting. I'll be very happy to keep you in contact, please, to share these advices.
Sure.
If I'm really concerned about being so many enzymes, if one batch of your assays is not as good as the other one, I'm really afraid of the system. Actually, I have no experience on it, so I'm really scared of seeing so many things in a single tube, and I really feel that I need to control each one of them. I hope it will be useful to see specific activity of each one. We will have to define the activity, the unit. So there is little things. There is, I think, little information about that. So, well, in a way, I'm also excited to contribute with this. So it will be something original, I hope. But I don't mind being less original if I have something more reliable from anybody, please.
It takes a lot of time to troubleshoot as well if something's going wrong. This is basically what Smith has had a bit of a nightmare of a few months trying to troubleshoot some of these reactions, because you've got, like, if you get a contamination or something, you've got so many enzymes you need to repurify and all the buffers to make up again. And it's very different. And even setting up the reactions, it's a lot of stuff to kind of put together. So it's not something you can just kind of, it's very laborious to do large scale experiments with it.
Maybe we can help each other a lot here, because if you go to the most basic physical principle of each one of these proteins binding DNA, that you must find a quencher or thermophoresis, some biophysical means of measuring it. And once you have a good batch, it should be regular. It should, the addition of every part should behave the same. I don't have a facility for thermophoresis, for instance, but it would be much simpler. So maybe at the beginning, until we have a user-friendly, a laboratory-friendly, a low-tech friendly means to characterize each one of the enzymes, maybe we can resort to a lot of help from better equipped laboratories. So if somebody in the room has thermophoresis, for instance, or SPR, plasmon resonance, I would be
We
very
have
happy.
plasmon resonance.
Yeah, we have
Yeah.
plasma in KEB, but not, I don't know about the other one. I don't know about thermophoresis, but we definitely have people. We have a whole group that are working quite extensively on SPR.
So we're going to try with this simple fluorescence quenching and keep you posted for help, please.
Yeah, I was just mentioning in the chat we could start an RPA thread on the forum to exchange tips as we go along. I mean, our other approach is just to kind of not use RPA and sort of dial it back to a simpler system for the typhoid projects. That we're not using RPA for COVID, but for typhoid. So SMIT is exploring some other options using the same enzymes, but fewer of them.
I will stop sharing.
Well, we've got loads of time to discuss it if anyone has any other project updates or if they have any other questions regarding any other work they're doing. So we can just open the floor to everyone now.
Maybe we can talk a little more about sort of IQRPA in general, like your experience with it. I've heard both good and bad things, like also a lot of false positives or not, or Jenny, when you were saying you guys have problems, it's there's some kind of contamination, everything is getting positive, and you're also measuring it with sort of lateral flow usually.
Yeah, well, it's the classic isothermal amplification issues. We haven't seen anything particularly different between RPA and LAMP. It's just a lot of non-specific, and then trying to find out if it is actually non-specific or if it is contamination has been SMIT's challenge over the last. I know Smitter and Akash has been exploring lateral flow. Maybe he can speak to that. We've been doing RPA coupled with CAS12. So, but yeah, Smitter can maybe update on her thoughts, having spent quite a few months now, getting on for a year now, focused on RPA.
After optimizing, I went through a lot of steps of optimizing in-house RPA. And pretty much when I was satisfied with these optimization, I started having contamination issue. And as you said, there are 4 enzymes, and one of them is very demanding in terms of its concentration. Going over 3 times of purification, I still couldn't get over contamination, but I'm happy to share all the optimization I have done so far if it could be useful for anyone. But be aware of contamination. RPA has, like as you said, it can detect 1 copy per microliter. So contamination from is a problem. Yeah, and I have been doing real-time RPA monitoring with a dye, and EVA green seemed to have worked better than cyber green in this case, which was helpful to optimize. And I've also, so the main problem with RPA what I faced was reproducibility of when the amplification starts, because it's so viscous and there are so many ingredients. It's very hard to get reproducibility in terms of the amplification or when the amplification starts, and it essentially also comes down to how pure can you purify a protein. In our case, we just had nickel NTA purification. If you can add another column of anion exchange later, could improve this timing. So it's good to also keep in mind there's a lot of reproducibility issue, but this issue is not with the commercial kit. It's pretty much with the homemade mix, and I'm not sure how to solve this. But setting pH 8 and above seem to solve a lot of my problems, and working temperature of 39 degree and not 37 degree also seem to solve a lot of problems. So it's good to also test a few more temperature range where you would get this reproducibility with RPA. But to be on a positive side, RPA is wonderful, but takes a lot of effort. If there's a good system to set it up, it's worth, I think, many other systems, as I think.
Yeah, and we have collaborators in South Africa who've also been working with RPA, although mostly so far with the twist reagents. But even with that, the problems with kind of viscosity and precipitation, having to mix the samples really well, are very pronounced. Like the difference between having samples that are well mixed and samples that you've just pipetted and not mixed is kind of night and day. And actually Twist sells, part of their equipment is equipment that just keeps mixing the samples while you're doing the RPA reaction. So they've obviously also identified that. And Smith was finding, like, more so than with the Twist RPX, which is reasonably viscous and still needs mixing, if you got homemade proteins, because there's so much in the mixture, they're likely the precipitation challenges. And that's where she did a lot of optimization on the pH and kind of concentrations of stuff to just prevent. And again, it might be, like she just said, if they're much purer proteins, you might not have those issues because they probably are closer to the purity of what's in the Twist DX. But precipitation is a key challenge. And so if you haven't got anything that's actually actively mixing the sample, I think that's probably adding to the variability as well, because the whole thing is kind of sort of the dynamics of how everything's working. I mean, you put a crowding agent in, right? So you're trying to constrain the sort of physical diffusion of the different parts of the reaction. And so it's a lot in terms of that, like, it doesn't just operate like normal diffusion in a PCR reaction. So it's much more sensitive to mixing, and it's much more sensitive to additives that could not only affect the chemistry, but also affect the physics of how the mixtures kind of diffusing. And that's an extra challenge, I think, because we don't see that with LAMP or any of the other ones. I mean, none of those, you actively adding crowding agents or PEG or whatever. So and we also oh, sorry, Rachel, you go ahead.
I was just going to say, for instance, if you had 3 reactions side by side on your real-time setup with the EVA or the Cyber dye, do you see all 3 coming up clustered, or is it really like just spread out for each reaction?
So it's spread out for each reaction, and as I said, reproducibility is a problem. But why I was saying it's better was once it reaches the saturation phase where the signal saturates, with Cyber Green, the signal seemed to go down, and there seemed to be some inhibitory, whereas with EVA it was pretty much flat saturation curve. It's very difficult to understand why something that has amplified would go low in signal, and it doesn't seem like photo bleaching or any such effects. It is some inhibitory effect where the template might be separating out to single strand. It's not clear why that's happening, but that's why I said EVA Green seemed to be better, because it eventually depends on where are you catching your signal, like where are you in the saturation curve when you are looking at the fluorescence or anything. So.
But I mean, you could just have an endpoint readout where
yes, but you have to be specific if it's going down, even when it hits us. Yeah, right. So you have to be so precise to hit the peak of the saturation curve.
Yeah.
Akash, do you have anything to add on lateral flow?
Yeah, it's actually what I'm doing now. So I'm working with the Millennial strips at the moment. We've got a decent supplier in the UK for those. Again, initially we were getting all the stuff from Twist, but as Jenny said, like the supply lines are so slow at the moment. I had to reach out and find someone in the UK who would ship not the RPA mixes, but the Twist strips to us at least. At the moment, we managed to get a 1-pot RPA Cas12 assay working, and I'm in the process of trying to convert that so that it works with the Millennial strips. there's a whole host of problems that you kind of dive into in terms of optimizing the reporter concentrations for these strips, to looking at the viscosity, as kind of previously mentioned, that the RPA mix kind of adds into the mix, because the speed at which these liquids migrate up the strips has an effect on kind of the different binding stuff and that's happening on there. So I'm in the process of trying to optimize that so that we get this one pot assay working on these strips, but it is a lot of kind of fiddly work and playing around and minute things, because the way these at least the TwistDX ones work with the CRISPR reactions are far from ideal in terms of a yes/no detection, because just the way that the strips work.
Do you want to expand a bit on how they work? Because I'm not sure.
I think I could talk for days about these things. But basically, the way that these, without like having figures behind me, it's like the way that these strips work is that they were initially used to detect amplicons as a result of PCR. So you'd use one primer with a FAM probe on one end, one probe with the biotin probe, and then when you've got your final results, you get a double-stranded duplex DNA with a FAM on one end, a biotin on the other end. And the way that the strip works is that it has two capture lines, as do many lateral flows. One capture line for the nanoparticles that run up the strip, and another capture line for the biotin probe. So if your probe has successfully been generated, what happens is the FAM on these double-stranded FAM biotin regions will capture the nanoparticle, and then you'll get a line forming at your test line. What happens when you're running the CRISPR reactions on these, and a few of the Sherlock papers have done this. I think a few of the detector papers that Jennifer Doudna is working with in the Sherlock stuff that like is coming out of the Broad and MIT, is that they've kind of used these strips, but kind of not fit for purpose in that they're using single-stranded fluorophore, so FAM and biotin reporters, and then trying to assay for the cleavage of these reporters. So from their original way, the test line and the control line have been switched around. However, because the initial control line bound only the magnetic beads without any other kind of separation, there is this weird kind of like game that you have to play to try and optimize it for purpose. Yeah, it's hard to explain without having all the pictures right in front of me.
Can I ask why you want to do this? Like, I understand why Doudna wants to do it because she has a patent and she makes money off of it. But to me, it sounds like the most insane way to detect the lamp reaction anybody's ever thought of. So
Yes. So there are probably other methods of running lateral flow on these things in terms of generating new different lateral flow assays. The reason kind of I'm working on this side of things is with the low-cost viral diagnostic project that I'm on, we're just trying to generate a few different outputs. And basically, since the project started, all these different methods have came out, and these Millennial strips were something that got published. I can remember 2 years ago now. So we started working with those, and now we've gotten to a point where coronavirus hit, we got kicked out of the lab for a while. There's not been like coherent lab time, and it's kind of got to the point where I'm trying to make, as an end goal, a lyophilized mix that works with these strips. But they're not the ideal situation. There are probably labs around the world / I know that MoLogic, the small lateral flow company, is definitely working with a few of the guys from Sherlock to try and develop new lateral flow strips, because these ones aren't fit for purpose. But just for my interest, it's a case of I don't have all the capabilities in lab to generate my own lateral flow strips. So I've kind of been adapting CAS reactions to work with these systems with the aim of just trying to couple these RPA CAS in one pot. which is kind of where the novelty of the stuff that I've been doing in terms of just generating these things in one pot instead of having this segmented workflow. Hope that makes a little bit more sense, but it is crazy.
At least everything is in more or less the same temperature. You don't need also like many temperatures. That makes a little bit more sense, but it's still a lot of enzymes that need to work for this thing to work.
No, definitely. At least RPA is so many enzymes. Yeah, initially the work that I was doing was trying to kind of see how we can boost the sensitivity of just Cas reactions by themselves. My old postdoc, Charlie, who sent a lot of plasmids around, was working on this kind of stuff, and this is kind of like my legacy project for him in terms of just I want to get his work done, published to like a decent standard, and then I can go back to fiddling around with Cas reactions myself.
I guess even if it's in one pot, both Guy and I are more comfortable if once the reaction is done, you just never open a tube. So when you go to put something on your lateral flow device, and I'll say from my experience, you should always try and test more than one dilution when you load the lateral flow thing, especially if you have these really viscous reactions. But if you could have some sort of fluorescent endpoint readout, I would be much more happy with it as an assay that could get into like resource strap settings and things like that. I understand that if you want to do something fun with what you have, but yeah, the dipsticks, we used to call them, it's just a contamination waiting to happen. you change your lab coat, you take your lab coat and leave it in the room, right, before you go out. That's what I would recommend. I also, it's amazing, these micro droplets. There are just so many molecules in them. At least they're less stable if it's just the two primers. Easier to wipe away with some bleach, but
yeah, no, I remember when I started doing the lateral flow, like the isothermal amplification stuff, like 2 years ago. I did like a couple of reactions and started getting weird contamination issues, and I just was like, at that point, looked at Charlie and was like, "You can do that. I'm going to focus on the Cas stuff that isn't going to make my life like a nightmare for the next 2 years." But he's gone now, so it's my turn to kind of take up that baton and see what happens. Yeah, at the moment we've got it working, so we get these endpoint fluorescent reads. This lateral flow thing is just kind of like another stick on top of that. I don't think it's necessary. It's just like a nice little output to have. At least for my thesis, I'm just like, look, it works on paper. It's not the most practical solution, but it works. So yeah.
How much DNA does RPA make? Does somebody know off the top of their head?
Sorry, that was a bit faint. I didn't hear that.
How much DNA does an RPA reaction make? Do we know more or less?
I probably need to quantify it, but yeah, no, I haven't done any quantification to work that out, to be honest, from my head.
Well, the bands on Daniel's gel that he showed probably were about the same intensity as one of the marker lane bands, so maybe 100 nanograms or something that was loaded. But that's probably not the whole reaction.
No, but those were PCRs.
Oh, those weren't the RPA, those were the PCRs, sorry.
Yeah.
We've also got an RPA presentation coming up from a team in Thailand who've been working with the Broad on a Cas13 RPA Cas13 assay. So we'll have more RPA chats, I'm sure, when they present and figure out. Unfortunately, it was a bit short notice for them and also quite late today, but next time around.
Yeah, and we'll give notice for when they are presenting. I don't know if we've sorted out a date yet, Jenny.
No, well, I think they're going to put forward a flash talk for the symposium, and then we should arrange a longer Q&A session at a later sort of standard meeting.
Perfect. Well, if there isn't any further discussion, I will highlight our symposium again. We have a Google form circulating, which I will put in the chat right now, which allows you to register for the symposium. It is on March 17th at 4 p.m. GMT time. So I'm sure that is ranging time across morning on the other side of the world. We are looking to have several flash presentations, some lightning talks. So I think we're aiming for about 5 minutes from everyone on those, and only a very short Q&A after that. We're looking at a few keynote presentations as well, and some breakout rooms for extended discussion. So we're really looking forward to this. So yeah, definitely register with that form and highlight if you're interested in giving a quick presentation. As well, if you're interested in volunteering with us, please do send me a message. We're always looking for someone to help coordinate the social media or facilitating a breakout room. That would be very helpful. So if I don't have anything else, Jenny, do you have anything to add?
No, I don't think so. Well, we're hoping, so we haven't confirmed that we'll try and do like a start and end keynote of a slightly longer talk. And thanks to Fernand for putting us in touch with Pharma Co-op, who we haven't fully confirmed, but we're hoping they'll do one of those talks. They're a company in Argentina who have reclaimed company, is that the term that Fernand, that they've basically bought a pharmaceutical company. The employees of a pharmaceutical company bought the company, and they're now like working on just sort of what they were doing before with pharmaceuticals, but they've also collaborated with a local biotech and a local university in Argentina to produce an antigen test. And so they can talk a bit about how that worked, the process, and maybe interesting for those of you who are looking more at the kind of from like getting your lab test out of the lab and into production. I think it could be a nice story between these three actors that kind of, and they have a very kind of mission-driven ethos from their website, which they'll tell us about. I don't know, Fernand, if you've got anything to add. You've had more conversations with them, I think.
No, not more. Just that was a very old company, like 80 years, doing pharmaceuticals, and then when they broke, all the people, the community took the company because it's permitted in the law in Argentina, and it's a very interesting story because they work on as a form of a cooperative in pharma. So it's very interesting.
Oh.
So we hope to see you for that. We have got a few more minutes left. If anyone's got any very quick kind of highlighted project updates, Guy, you've got your.
No, I have a few like general stuff. I'd really like to hear Chinna about his kind of off-the-shelf isothermal heating solution at some point. And then for these things, so the last one, I know it was recorded, but it's not up on the thing. And then also in general, right, I'm part of the Reclone, and I signed up for, so I'm not, is there a way to get an email about these things, right? Like somehow.
Yeah, so we're
every time I get everything that happened in the Reclone forum, like exactly the day after this all happened. So I'll miss it. Right?
Oh, I see, because you must be on the daily updates thing. So yeah,
or like
you can.
Weekly, I think I get one once a week.
Ah, okay. So you can change your notification settings to get the messages more frequently, which will probably help. If you log into the forum. But we can definitely do some kind of calendar invite that at least gets it in your calendar.
And then we should also probably save the chats from these things.
Mm-hmm.
That's the thing.
Definitely, yeah. I think the chat would be very helpful, and you can always refer
last time
back
I think
to.
it just got closed and then it all goes away. So somebody before they close the room should copy that somewhere.
Well, it's stored whenever you record a meeting. So you should have the chat from last time, I think, because it would have been stored in the recording zip file.
I do the transcript, so I can upload that.
Nice. Yeah, you can just copy and paste it into the end of the notes that are going on the forum maybe, and then we'll have the recording and the kind of like notes and chats transcripts. Would be great.
So I would like to maybe give a small update on the hardware side. Sorry, Smith, I supposed to write to you before updating here, but yeah, this is the device I'm trying to develop right now. It's an LCD screen that gives the temperature, and this is where the tube, the lamp tube goes. It's only for one tube, but as you can see here, it's just very handheld, and it will cost maybe anybody to build maybe $5 or so, depends on where you get your boards and where you get these boards. So it's based on this W1209 board. If anybody is hardware hacker here, they might recognize it's a $1 board, and for $1 you get this microcontroller, LCD, relay, and buttons, everything for $1, $1.5, along with the temperature sensor. So all you need to do is that having the access to a heating element and power source. So right now I'm powering everything from a mobile bank, but you can use mobile phone charger, anything. And for heating source, I'm using 3D printer extruder cartridge. So yeah, that's all I have right now. So I'm still waiting for one of the part, because if I send my working prototype, I won't have anything to work on. So I'm waiting for a part to arrive from the UK since last 2 weeks. I'm in Ireland, so we have terrible problem because of either Brexit or simply.
We've all got problems because of Brexit, Jenna. join the club.
No, I never had this problem. So what I would like to suggest to Smita is that I can send the parts list and I can send the 3D print files. Maybe if I don't know how much experienced you are, it's easy, fairly easy. You don't need any soldering or no programming. So it's just putting them together. So that's another important point in this device, that many devices you can see in the literature, they need at least Arduino programming or soldering or SMD part, but this doesn't require any of those. So this is also very, by the way, it is 1.5 or $1 because it's highly popular circuit to incubate the eggs. So people use heating element, tungsten filament lab as incubation source. That's it. They don't need anything else. So they connect it to the AC source. But when it comes to, my aim is to run this with a mobile bank or 5 volts, because this voltage source I think is most commonly available because of the smartphones or mobile phones. The problem is people use the AC source, the tungsten lamp. So you need to find a right heating material that is with that will not draw so much current, but at the same time provide the sufficient heating. So it will not be a perfect solution, but I think it's the least cost effective solution I found. Maybe there are others as well. It will not give out any reading, real-time readout. It just, you run the lamp assay or water assay and you take out. I think there are so many apps in development to do the colorimetric assay. So that's all from my side.
Nice. I see we've got 2 minutes to go. I don't know. Unfortunately, I had to miss some of the last meeting. Did Chiara mention the plate auto-induction, Smita, or anyone who was around the last meeting? No. All right. So we'll put it on the Reclone forum. Chiara, for those who don't know, started a new job this week, which is very sad for the Open Bioeconomy Lab, but great for her, and I hope she's having a great time. But before she went, she did wrap up some experiments where basically we don't have ready access to a centrifuge in Cameroon that can do large volume cultures, and so it's tedious basically to pellet cells after putting them overnight. And we've been testing auto-induction media versus IPTG, and we found auto-induction media, at least we haven't tested with the RPA enzymes, but at least for polymerases and others, is really nice. And you can use auto-induction media with standard lactose, or Chiara also tested using whey produced from milk, which does work, but is a bit of a faff to produce it, or just adding milk, which does work, but your optical densities are thrown completely off. so that wasn't ideal. So she now tried making auto-induction media in an agar form and just growing the cells overnight on an auto-induction plate, and then harvesting the cells the next morning. And I can quickly, we've got 1 minute to go, I'll just show you her video. This is her harvesting. Oh, hang on a minute, Zoom is making it difficult for me to press the play button. So I don't know if you can see that. That's a plate, a confluent plate of cells expressing open vent. That's a plate on LB. They don't seem to like LB so much, but they were plated very well on auto-induction. And then she's just using a razor blade there to harvest the cells from the Petri dish, and it comes off really nicely. Ta-da, it's very satisfying. And then you can basically scrape that straight into a tube and go forward as if you just pelleted the cells from your centrifuge. And the yields are pretty good. I mean, they're definitely comparable to LB and autoinduction media. The only thing that beat them was autoinduction media, so rarely rich media in a baffled flask. That did get high yields, but only by like 20%. So yeah, so if you don't want to faff around with centrifuging, you can, and you've got a T7 induction system, just pop it on a plate. And milk works. So she just made like basically autoinduction agar with just milk from the supermarket, and it got a really nice fat band on the SGS page gel. So if you want to save yourself some time, there's a top tip. She's put all the protocols together. Yeah, it's quite satisfying. Yeah. Instant gratification. Yeah, we'll post all the stuff to the Reclone forum soon, but I wanted to share that little mini update.
Lovely. Well, that is the end of our meeting today. It was so lovely seeing you all, and thank you, Danielle, for presenting. It was very interesting, and we'll definitely get an RPA thread up on the forum as well. So thank you, everyone, and hopefully see you all at the forum in 2 weeks. Thanks,
Bye.
everyone.
Bye, everyone.
Bye.
I just end it. Oh.